our Innovative Discovery series today. We are absolutely honored and very excited to have Dr. Melissa Brondy giving us a talk about molecular and genetic epidemiology of glioma. But before I introduce our speaker, let me go over some housekeeping issues. We continue extending CME credits for our tech talks and our ID series presentations. We will distribute the information via email to those that registered for today's event, but if you did not register prior to today's talk, you can still obtain CME code information from Deborah Rees, and her email address will be posted in the chat box today. Our next ID series is June 22nd. Dr. Anjana Bhatt from University of Delaware will talk about motor interventions for children with autism spectrum disorder. So you really do not want also to miss this presentation. So again, I'm extremely excited to introduce Dr. Brondy and to attend her presentation. Dr. Brondy is a chair of the Department of Epidemiology and Population Health at Standford University School of Medicine. She's also the co-director of the Standford Center for Population Health Science, as well as Associate Director of the Population Sciences at the Stanford Cancer Institute. She obtained a PhD in epidemiology from University of Texas from the School of Public Health in Houston, and she has been the PI on numerous NIH R1 grants over the years. She has a very impressive list of peer-reviewed publications, almost 400, and has written, participated in the writing of multiple book chapters. She's a teacher and a mentor for numerous clinical and research fellows, as well as graduate students. And she's part of multiple advisory boards, such as the NCI Board of Scientific Advisors, the Central Brain Tumor Registry for the United States, and many, many others. She has received multiple scientific awards and honors, but the one that I really like the most is the Best Boss Award from the University of Texas Anderson Cancer Center. And she's been invited all over the US and the world to give talks and lectures, several as keynote speaker. So we are really grateful for Dr. Brondy to spend time with us this afternoon. And thank you so much again.
Thank you so much. It's a pleasure to be here, and I'm really honored to have been invited to participate in this Grand Rounds. So I'm going to present, and then I want to leave time for questions. And I also want to make sure that people understand about glioma and the difficulty of trying to study this disease. And I think that's why I've spent my entire career doing that. Claudine, can you see my screen as I advance it? Okay.
Yes.
So I'm going to start out, and I want to give an overview of descriptive epidemiology of glioma, as well as talking about the risk factors and some of the studies that I've been involved in, which we've called the gliogene studies, because we've been trying to identify genes that are associated with this rare type of brain tumor. So I'm going to talk about our family studies. We have new data, so I really wanted that just was published, so I will talk about that, and then also our future directions and studies that we're planning. So just to give you an idea about brain tumors, they account for about 1 to 2% of all cancers. So remember, it's a very rare cancer. And the number I'm showing here on the right side is that there's 88,190 or so brain tumors that were basically diagnosed in 2022, but the numbers really haven't increased very much. This includes all types of brain tumors, both benign and malignant. Of the malignant type, which we call mostly the type that I study are called gliomas, it accounts for about 26,000 cases per year. And the rest of those are non-malignant, and they're often the most common type are meningiomas, which are a benign type of brain tumor and account for the majority of the benign types. And remember, the survival is really, really poor. The 5-year survival for a malignant brain tumor is about 35%, but for GBM, the glioblastoma type, the survival is even rare. About 9 months is the median, and now with a little bit of some treatment options, it could go up to about 18 months. But it's a rare that that people affected with this disease survive very long. So just to address a little bit right on this histogram, the distribution here, there's a bimodal. We've thought there's a bimodal distribution between childhood brain tumors and adult brain tumors, and they occur early on in a child's life, and then again they peak at about age 55 or so in adults. So you can see the difference here. In the histologies, the type of brain tumors we see in children are also very different than the ones we see in adults. I'm going to focus today on adult brain tumors because I've studied childhood brain tumors, but the work I'm presenting today is primarily in adults. And you can see that the difference in the incidence per 100,000, and for the malignant type in children, it's about 3.73 per 100,000. For those under 14, it's a little lower if you include those the young adults. And then when we look at the adults, it's about 8.76 for malignant brain tumors per 100,000 population. So here it is. I just wanted to show, this is a little older, but I'm just bringing up one point here. This accompanied an article that was published in the Journal of the National Cancer Institute. And this was just looking at SEER data, and there was a concern in the early 2020s or so that cell phones cell phone use the brain tumors were increasing with cell phone use. And the SEER data were published in 27. This was from 2017, and you can see that if there was an increase related to cell phone use, that we would start to see increases over time because everybody uses them. But this is just looking at time trends from 1975 through 2014, and you can see that the rates for both males and females are fairly flat. And there has not been any spikes or increases in the development of brain tumors over time. So people always ask me this question, well, what about cell phones? Are they related to brain tumors or not? And I want to say that I don't think that the evidence is very strong, and that if we did, or is strong, and if we did see it, we would be seeing changes in the incidence data. So what do we know about glioma and the epidemiology of this disease? Glioma accounts for 80% of the primary malignant brain tumors, and approximately 23,000, as I mentioned earlier, 880 people will be diagnosed in 2022. And I should have added the 2023 data that's projected, but it's about the same. There has not been a major increase. But the point I want to make with the next, the mortality, is that about 16,000, almost 17,000 will die from the disease, which is not too different than the incidence. And that's what makes this so difficult, is that over so many years where many cancers we've seen the prognosis and survival has improved because of better treatment, we have not really seen this in with brain tumors. And partly, and I read an article today in Nature that there is some new treatment. Well, it's not necessarily treatment, but a way to modify opening the blood-brain barrier so that the treatment, the chemotherapy, could get across the blood-brain barrier. And that's part of the problem is because of the blood-brain barrier that it's really difficult to treat this disease. It's not like we're treating lung cancer or colon cancer or another type of cancer where it's accessible, and the treatments work much better for diseases like that than they do for glioma. And as I mentioned, the median survival of is about 1 year, and 90% or so will die within 3 years. What's really been of interest to me is that we've seen very few cases occurring in underrepresented minorities and Blacks and Hispanic Latino populations, and it's so much more common. And we have a study right now looking at race, ethnic differences in glioma, and it's taken us an exorbitant amount of time and contacting many different institutions to try to get access to samples from minority populations populations. We had a spore grant that we've collected these, and we were lucky to get about 500 matched samples among Latinos and African Americans. So there's also a difference, and I think you could have seen that. I didn't mention it on the slide where we were looking at time trends, but the male to female ratio is about 1.4 to 1. And again, the mean age of the disease, of getting the disease, brain tumors, is about 55. And this is the most common solid tumor in children. So what about the risk factors? So we know very little about risk factors except for radiation exposure, and I'll give you an example of that. But a lot of the research has come from childhood cancer survivors, where they have been treated with radiation and they developed a brain tumor later. And I'm going to show you an example in a minute. We've seen in some of our case control studies that respiratory allergies are protective, aspirin use, and this is baby aspirin. And I'm going to show some data. I just wanted to list some of these things that I'm going to come back to. And again, genetic predisposition, which I've spent my entire career looking at familial predisposition and genes that might predict this risk in families, and it occurs in about 5% of the families. And the point I want to make here about genetic predisposition is developing, if you have a family history of a brain tumor, your risk is, this occurs in about 5% of all patients with glioma, but it's the same in all cancers. When we think about the attributable risk, or what proportion can we account to genetic predisposition of a disease, breast cancer is about 5 to 10% with BRCA1 and 2, and many of the other new genes that we've identified. In all cancers, that when we look at the genetic inheritability, it's about 5 to 10% of those cases. We've also identified brain tumors are more common if you've had some of these rare genetic syndromes, like Li-Fraumeni cancer family syndrome that's been related to p53 mutations. Patients that have neurofibromatosis, both type 1 and 2, have also been predisposed to developing a brain tumor. So this is such an interesting story. And if anybody feels like they want to spend, I don't know, maybe an hour or less watching this video, you can find it on YouTube. And I don't have the site, but if you just search on the ringworm children, you can see that this video. And what's so interesting, so when there was a major migration into Israel from many different European countries right after the war, and these children were developing ringworm, which is a fungal infection on the scalp. So the doctors in Israel contacted other physicians around the world, and they said, oh, well, if you treat them with radiation, you'll get rid of the fungal infection. But I just want to point out, too, radiation was used for so many different things way back when. For those of you who are a little older might remember when we went to a Buster Brown shoe store, that they would take a picture. You could put your foot in one of these little things to do the correct sizing of your foot, and you could see some of the bones. And I mean, it was really crazy. They treated acne. There was stories about young boys who were given radiation so that they could continue to sing with high-pitched voice. So radiation was, like, in the 50s or so, was really something that was used for treating many different kinds of things. So what happened is that these children would come in for treatment. There was families, kids, because they, it was kind of infectious in a sense, had this ringworm. And so they brought the kids in. The parents, I don't think, really knew what was going on, but this was the treatment. And so they came in for several days or so, and you can see these little skull caps. They'd sit the kids in one of these chairs. They would zap them. And over the years, like 40 or so years later, they started identifying that these kids that were treated for their ringworm had started to develop brain tumors. And so the state of Israel developed a cohort to follow these, and they compensated them for what was done. But this was a natural experiment where we were unfortunately able to see the effect and the dose of the radiation and what happened to these children that many years later that they were diagnosed with brain tumors. So I mentioned the respiratory allergies, and so we conducted a case control study of about 13 different sites around the world, and you can see right here these were the study sites, and this included the whole country of Denmark, Israel, Sweden, and parts of the UK were also included, and then there were many sites in the United States. So the top one is all glioma, and the bottom is we partitioned the data and subset just those that had a GBM, which is the glioblastoma that I mentioned. So just a little bit of background and the hypothesis and biology surrounding why respiratory allergies is that they, what we identified, and if you look at these forest plots, you can see that there was about a 30% reduction in risk for those that had respiratory allergies. We looked at all allergies, but it was really the respiratory allergies that that showed the risk reduction, and so you can see in both of these the odds ratio, and what we did is we conducted a random effects model, and you can see that for most of these sites, what was interesting is, and these are the numbers of cases and controls that were in each, and what we did is we weighted the odds ratio according to how many cases and controls were in there. But a little bit of background is that we did here identify a 30% risk, and this is similar to what was published in several other studies. There haven't been many, but there has been other cancers, epi studies of other cancers where they've seen that respiratory allergies are protective. So what's the biologic mechanism? That it causes, these allergies cause a heightened immunosurveillance that reduces tumor growth. And there was some research done at UCSF where they were looking at IgE antibodies against these allergens to see if they displayed some cross-reactivity to brain tumor antigens, and they identified that they did. And individuals who had stronger allergic responses were more likely to expel some of these environmental toxins or carcinogens that reduce their risk of developing a brain tumor. So again, we were interested in like COX-2 inhibitors and aspirin, and that for similar reasons, we were wondering whether or not aspirin use could reduce risk. And so we asked about, different types of COX-2 inhibitors, and we also asked about aspirin, but the aspirin we asked about was baby aspirin. So this is the low dose baby aspirin. And so this first on the left, the first forest plots includes, we did a meta-analysis of other studies that have published aspirin use, and this would be regular or never. But when we did our analysis from our glioma case control study, we removed the cases that came from Europe, because in Europe they do not recommend baby aspirin be used for cardiovascular types of prevention. And now, according to the U.S. Preventative Health Task Force last year, they also said that they didn't think that it was really protective for cardiovascular prevention. So, but until then, it has been, and a lot of people still take baby aspirin. And you can see here that, again, there was of all the studies, about a 0.63 odds ratio that showed protection in our random effects model. When we looked at long-term use or versus never aspirin use, we also identified, let me go back, that there was a 0.64 odds ratio that showed a protection. So what we wanted to do then was to look at start looking at our data in a little bit way to see if how it really affected glioma risk. And we compared just our GICC data to by years of use and compared with those who never used. And what I wanted to point out, is that the odds ratio was significantly reduced when you compared it to those who didn't have, who never took it. And you can see that if you, for those who took aspirin for more than 10 years, the risk was reduced to 0.46, and there was a dose response relationship with aspirin use. But, it's really hard for us to tell people to take it when you don't know risk, but I think this is important. And we tried to do a study. I was collaborating with some of my colleagues who were at the University of Pittsburgh and now at UCSF to try to replicate this in an animal model. And we had some success and published a paper in the, I think it was in Cancer Research or one of the ACCR journals showing this protection. So the next point I wanted to make is, just to show some of these risk factors, is that there are hereditary syndromes and genes that have been associated that reduce that brain tumors are part of those syndromes. And as I mentioned, and these are all very rare syndromes, is that NF1 and NF2 have been gliomas have occurred in patients that have these syndromes. And again, P53 and Leaf-Romani has been identified, and brain tumors are the second or third most common type of cancers in those families outside of sarcomas, breast cancers, but Leaf-Romani is a cancer family syndrome with multiple cancers. It's been also seen in other types of rare syndromes. So, what about everything else that we keep hearing about that are mostly unproven cases? We've seen studies of head trauma, dietary nitrite consumption. There have been studies of both tobacco and in children, and so many of these have not been proven. We tried to look at diagnostic ionizing radiation, which is different than what I showed you, and just to see if people who had X-rays to the head area way prior to developing the brain tumor, if they were associated. Problem is, people don't know the difference between what type of X-ray they had, if it was an MRI or a CT. So it was really hard to evaluate that. For childhood brain tumors, there's been a lot of studies that have looked at many different kinds of things, including parental tobacco, residential EMF, and air pollutants, and just many, many types of things. And nothing has really panned out. It's probably because of the rarity of this disease and not having enough cases to be able to evaluate this in a systematic way with enough power. So I've spent a lot of time looking at genes that cause brain tumors, and these were several types of studies. We started looking at families, and that's what I'm going to talk about next. And then we also conducted through those sites that I showed you, case control studies to look at populations and associations with GWAS, and we did publish the largest GWAS study that was ever done, and found 25 SNPs that are associated with glioma, and we're continuing to work with those data. But I'm going to focus now on families, where we're looking at rare variants and single genes that could account for development and etiology of brain tumors. So what was our reason? And when we started doing this work many years ago, our rationale to study families is that we thought that this would provide insights to important genes that could be related to not only families, but also sporadic cases that would have not been related to a familial predisposition. And the reason we had to really start this by a large consortium is because of the rarity of the disease. So these are the sites that were included in what we called the GlioGene International Consortium. And as I mentioned, we started it as we put this together as a family study, and then we expanded it to do a case control study where we were able to then look at associations, and some of the case control data that the epi data I showed you came from that study. So our original aims when we started was to screen these sites, everybody who came into these cancer centers. And these were the largest cancer centers where brain tumor patients came to around the United States, and then the several countries in Europe and Israel. And we collected families that reported to us that they had at least 2 family members that were still living. So remember, I showed you earlier that the mortality is short, and so it was really difficult for us to identify multiple living individuals in a family that had a glioma. So with the 15,000, we estimated that maybe 3% would fit this criteria. And at that time, after the first study was completed, we ended up with about 400 high-risk families, but many of these were, and I'll show you some of the data. So we identified mutations with what we called familial glioma. And since the GlioGene study ended, we have an ongoing grant right now with just a few sites, and we're collecting cases through physicians. So if anybody who is a neuro-oncologist or a genetic counselor and they see patients that have that fit our criteria, please refer them to us, and we have a website. But we're also collecting people through when people search on Google, and we have a whole bunch of search terms, we're identifying quite a few people through that mechanism. And I have a student who's working on analyzing those data right now. And so hopefully we can show how well you can recruit through the internet. And we're also, we have a nice film. So if you go onto our website, you'll see the video that we did as part of our recruitment effort. So this is an example of a sample pedigree. And when we recruit families, we ask information about all of their relatives, and we get cancer history on each of those to see who had developed a brain tumor. In the earlier study, we were collecting all the path reports and medical records to be able to validate all of those. Currently, we are collecting blood samples, we're getting tumor samples, and then we get we try to get a path report to make sure that we can validate the type of brain tumor they had. Okay, so just a little bit about some of our numbers of all the families sequenced. They had to have had at least 2 verified gliomas. And what was difficult, and that this analysis is all in the blood or in the germline, or we also collect buccal samples or spit samples to be able to extract DNA. But one of the things I want to point out is that technology changed so much over the years, and technology has driven much of this research that we've done, because when we first started, we were doing linkage analysis with an array that did not have very many genes on it. And now we're doing whole genome sequencing, and we're in the process of implementing for the families that we haven't been able to identify genes. We're doing what we call long reads, which is a new type of whole genome sequencing where it's even more, the technology is even better. It's a lot more expensive, but for right now, we're doing some pilot work to be able to do those analyses. So right now we've sequenced about 332 cases from 314 families. And of those, we had 2 affected. There was about 40%. Another 42% had 3, and those that had more than 4 affected, there was about 18%. And what we were looking for, and my colleague who is doing a lot of these analysis was actually my doctoral student when we were both at Baylor, and this was some of the work he did, but now he's at Rady Children's, and he's still working with us on these families. So we do very stringent variant filtering and prioritizing of these genes. And we look for protein affecting variants, looking for splice sites and those that have non-synonymous mutations. We go through another filter looking at minor allele frequencies, and we look at those that were in the 1,000 genomes, looking at other types of variants. And this research, most of the sequencing was done at the Baylor Human Genome Sequencing Center, and they have a huge database that we're able to screen to see what we would have expected of these types of mutations in a population that does not have brain tumors. Also, we prioritize according to known cancer genes. We've identified through our SNP analysis and through other work that we've done over the years that many of the DNA damage and repair mutations and other candidate genes also are important in the etiology of glioma. So we've looked at these and looking at highly deleterious mutations as well. So what we identified from our initial study was, and this was published in 2015 in the JNCI, from the first set of analyses, we identified a gene called POT1. POT1 is encodes for a component of the sheltering complex family of genes, and it's involved in regulating telomere length and protection maintenance. What's really interesting about glioma is that the telomeres are lengthened, not shortened. So in many cancers, the telomeres are shorter, and many diseases, it's involved in aging and other types of things. So when we published this, we measured the telomere length in our cases to identify that they were longer. And so this is one of the first genes to be found in mutated in human, the telomeric structure found to be mutated. When we published this, there was another study that came out at the same time with familial melanoma, and they also identified POT1 mutations. And right here are all the different variants that were identified in POT1. These in red are the ones we identified, and these others were those that were identified in some of the other studies that were published. And so it was also discovered in chronic lymphocytic leukemia before we did in the blood. And so we were fairly confident that this is an important gene. Since we reported this, it has been included on the panels of genetic mutation panels when people are tested. So this was the first set of families that we published. And what we did in these is when the probands, the ones that are in black, are what we consider those are the ones that we were able to sample with a glioma. Well, and when you see like this number, this was the variant that was identified in this particular person. So in these families that we had blood on, we did Sanger sequencing to see who in the family also carried that mutation. And if there's a WT, that means they were wild type for it, but they were tested. These others that have it have the same mutation, and you can see that here with the notation. We are now in the process. So when we conducted this analysis, I was at the time working at MD Anderson, and we didn't have access to continue following the families. Now we are able to follow them. We've gotten permission, and we are now recontacting all of the families that we've identified with some of these mutations so that we can find out what the risk is for those others in the family that might be carrying that gene, if they might have developed a brain tumor or others, so that we can start to tell people what their risk is, because one of the all of the questions we're asked is, what happens to my children, what happens to my siblings? So we want to be able to tell them what the risk is, especially in genetic counseling and genetic counselors to do this, what the risk is of developing a glioma if they do have a POT1 mutation or even other types of mutations. So I'm not a typical epidemiologist. I mean, I really am, and this is my love. But when I moved from MD Anderson to Baylor, I was obviously acquainted with the Human Genome Sequencing Center. I had developed some incredible collaborations, and one of the collaborations I developed was with the neurobiologist Ben Deneen, who had developed a incredible CRISPR model. And so we talked, and I said, we're starting to identify these genes, but I want to know whether or not we can replicate and see what the function is of these particular genes in your CRISPR model. So this was the picture of some work that was, I think this was the picture that was on the cover of Cancer Research that of the results of some of the POT1 data that I'm going to show you. And this was the cover of that. So just to show that about there's more than 3 million cancer mutations, and about 5,000 of these are functionally annotated so far. And so this is just a little bit of the work that we published. We have an ongoing grant with Dr. Deneen looking at other types of mutations that we've identified in our families. And so this is the triple. He has a triple CRISPR model, an animal model, where we then take the gene of interest, and it is then put into this animal model. And then we look at multiple things about whether or not there's proliferation, whether or not it's expressed. in the animal, and also looking at survival, but also function of this particular gene. So we used the POT1 G95C cells to increase the tumor sphere frequency, and this is shown in this panel where we were able to show all of these different variants compared to the controls. And there's where we were looking at the cell numbers, and also you can see here on panel C where we were able to show statistical significance in the cases. And the one that was most significant here compared to the others was this 95C mutation. And so we were also looking at the coding, and this is on panel G is just looking at the prefrontal cortex to see whether or not we could see this particular gene lighting up in the animals. And we use this BRDU staining method, and you can see that they were fairly, the proliferation and the count was much higher in those with the POT1 mutation. So with these proliferative effects of the POT1 G95C and this POT1 knockout, we were able to show that it was it was functional, and that we were able to show brain tumor growth in that animal, those animals. So let me just go back now and show you some of the work that was just published last week in Science Advances. And this was the additional sequencing work that we did, and we had 325 subjects. We were looking at known variants and other indels, and I showed you the way we were filtering. So these were the different we were looking at loss of function, or those that had highly deleterious variants, and those that might have had cryptic sites and different types of mutations that we were looking at. We looked at the 25 SNPs that we identified, and these were those that we did. Those that we identified that were novel was HERC2, and I'll tell you a little bit about this new gene that we've identified. And we've taken this particular gene, and we're now looking at it in the CRISPR model. But all of the genes we identified that were new or novel, we were also trying to determine whether or not these are significant enough to put into an animal model, and that's how we've been working with our colleague Ben Doneen. And so this HERC2 seemed to be, and what we published was this was the most significant gene that we've identified in multiple families. So, you know, we always say that we're looking under the lamppost and trying to look at discovery cohort. So what we had for the discovery cohort was we worked with the Human Genome Center, and they're part of the they were sequencing many cases from the ERIC study that was part of their last grant. And so we had, looking at what we would have expected in a control cohort, we compared those that we identified. So we have identified these genes, particular genes, these cancer predisposition genes, and they were more significant compared to what we would have expected for these 7 cancer genes. So this is just a list of the families, and I just want to point out that we identified multiple families with these particular variances in HERC2, and these were the 5 different families that we identified, and this was more frequent than any of the other genes in these particular families. And these are very rare variants, those that you would not have expected to occur in a normal population. So what is HERC2? So as I just mentioned, it was the highest enrichment for deleterious variants compared to a control cohort, and it was mutated 6 times, 5 rare, and it predicted highly deleterious variants, and once as an internal copy gain. It's highly conserved throughout the human population, and we found it was marked selection against a loss of function. And it's extremely large, so it could still harbor harbor many variants in normal individuals, but we didn't see it in our comparison population. And it's possible a gliomogenesis gene. Nobody else has published this, and especially through our variant calling that we did, we found this to be significant. So I'm going to tell you a little bit more about what its function is. It regulates the ubiquitin-dependent retention of repair proteins on damaged chromosomes, and it affects the double-stranded break excision repair pathways. Remember, I told you a little bit about that, that the DNA repair pathway genes have been very important in brain tumors, as well as it modulates P53 activity. P53 has been identified in multiple brain tumor cases that we have. It's also, the SNP is also relevant, as well as this being involved in leaf-remaining cancer family syndrome. So the function made sense. It's may only act as a tumor suppressor in the context of the wild type P53. So we're still trying to investigate what normal levels may be needed to observe this tumorigenic potential in HER2 mutation. So hopefully with the work we're doing, we're going to be able to unravel this, mechanism and function. And it indirectly regulates the insulin growth factor pathway and cell cycle, and may be involved in immune function. And remember, we talked earlier about the role of immunity and brain tumors. And it's commonly mutated and focally deleted. Well, we found it in our glioma cases. So other observations, we identified this MYO7A, and it was mutated several times. CPGs accounted for 18 families, and these were some of those that had loss of function. We previously published, and this was in our first linkage study, this region. This was before we knew anything about any particular genes, and now we were able to identify what those genes were in these regions that we identified from linkage analysis. So we were kind of excited about that, because we weren't sure what that meant in our early families. And then we've identified several new types of genes, but in a small number, that can explain the etiology specifically for those families. So unfortunately, for most of the families, they look like that they're private mutations, and that we haven't seen them like a BRCA1 gene that we can say, okay, now we can test to see what your risk is because you have this particular gene. We're still identifying quite a few different genes that could be the causative gene for that particular family. And then we identified additional POT1 genes in the new families that we sequenced. So just to conclude, and I'm happy to answer questions, is that we know that the POT1 in CRISPR identified proliferative effects, and we were able to knock this out in the CRISPR model. We're continuing, as I mentioned, to collect new families. We're going to be doing additional sequencing in the next year, and hopefully with larger numbers, we'll be able to identify more of these genes. But I'm really interested in trying to understand the risks to other people that are in these families who may not be affected yet. And as I mentioned, we're conducting this CRISPR model work. So maybe I thought I had ended this, but so this was some of the results that we identified through the screening of those genes that I mentioned to you. And so this was trying to look at the function using a functional screening method. And this was a barcode method that we used to see whether or not these would be important to put into the CRISPR model. And here's where we looked at some of the loss of function and staining to see the role of these particular genes and those that we think could be important for future research. So some of the future studies that we're doing, as I mentioned, well, I didn't mention this, but we have a large study, and in fact, looking at the cases, the tumors. We took the tumors from many of the cases from the United States sites, and we're doing somatic and germline mutations and looking at subtypes so that we can begin to look at the etiology of and survival and outcome, looking at different mutations. And so we have a large study of over 2,000 cases with a large number of minority patients as well as Caucasian to look at outcomes in those families. And conducting functional analysis continues as well. So it takes It takes a lot of people to do this research, and these are my collaborators from around the world. And I want to thank specifically our families that have participated and allowed us to continue to do this research. So these are my collaborators, the different groups that I'm working with, and I'm happy to answer questions.
Thank you so much, Dr. Brondy, for this really enlightening presentation. Any questions from the audience? You can put question in the chat or open your microphone. Well, while people are thinking about questions, I have several actually.
Oh, good.
So I was wondering, did you find any geographic association with the glioma? I mean, you mentioned some of the countries that you are working with, but are there countries where there is really a much higher incidence in glioma, and that could be also related to genetic issues?
No. So, I mean, let me answer that in two ways. So there was the reason we worked with the countries we did is number one, Denmark and Sweden have registries. And they were also part of another study called the Interfund study. So they had already been collaborating with each other. So that's why we included them. So in terms of so that was the reason that we worked with those countries. The other question is, and one of my former postdocs who I continue to collaborate with, Quinn Ostrom, has done a lot of work of looking at SEER data, looking at the CB Trust data, and starting to identify where many of the, looking at national data to see if we see any clusters in areas that could be important. She had published a paper about a year or so ago looking at the mapping the patients, and she did find an increase, and I'd like to go back, we need to go back to that, showing where there's more radon exposure, which makes some sense. And I can't remember where she published that paper, but it was published about a year ago. And, we've also been looking at the distribution by race, ethnicity, and looking at ancestry and things like that. But in terms of the environmental exposures, that was the only study recently that we've been able to look at that had some interesting results. In terms of there's been studies of exposures along freeways. There was some studies by Peggy Tucker, and it wasn't just brain tumors, but she also looked at leukemias in California, and that, many of these occurred along freeways. So could be exposure to environmental diesel fumes and things like that. But those were all cluster types of analyses, just looking at spatial clustering.
That's really super interesting. Do you think there is some type of epigenetic a phenomenon where exposure, for example, to radon would trigger the function of the gene of this POT1 gene that you
I don't know about
identified?
POT1 so much, but if it's, I don't know how if radiation would induce that, but there could be other genes, maybe P53 and others where it could, maybe POT1. I mean, we haven't been able to look at that, but I need to ask, we should probably look at some of these a little bit more closely in that work that she did and where the clustering is. But, that was just, a one-off kind of analysis that she did. We're sort of trying to look at other things. We're taking real world data and trying to look at co-occurrences of different types of neurological diseases and see if there's any relationship. It's such a hard disease to study, and we keep trying to, look at this in so many different ways. I work very closely with the group at Duke, which is, where Quinn is, and Kyle Walsh. They're close collaborators. And then the Memorial Sloan Kettering, I still work with people at MD Anderson and Baylor and UCSF, and so we keep our connections, and our studies are still still ongoing.
Thank you. Mina, do you have a question?
Yes, thank you so much, Dr. Brondy. Amazing presentation. You might have mentioned this, and I might have missed it because of connection, so I apologize. But have you seen any differences in different races in terms of inheriting cancer genes?
Not really, I mean, because we do have minority cases that are in our family study. In terms of, the familial component, we have, I can't say that. We don't have enough. I mean, we have in our family study right now, we have over 300 cases. In our other studies that we have of the minority, we haven't looked at the family history in those, but we will as in the 500 more that we've collected. There have been some studies in Mexico, by the way, and but the interesting thing, and I presented this at the SNOW meeting, is that for those cases, and we published a couple of papers already, the survival in the African American cases and in the Hispanics, the Latinos, is better. So whatever that means, I don't know, but their survival is better than the Caucasians, and even those that have had standard of care treatment, once you account for that.
Oh, that's really interesting.
Mm-hmm.
That's intriguing.
Yeah, that's what we're pursuing now, and hopefully we'll have really much better data as soon as we finish this study, and we're hoping to get it done within the next 9 months or a year.
I don't know whom we are seeing here. Any other questions? Well, I have another question actually. So you said that there was like a bimodal in age, so children and then adults. But among adults, is there also more of a cluster among older adults or younger
I think
adults?
it starts around 50. We see 55 to 61. It's sort of, and then it declines some. But that's where the peak age is around 55, between 55 and 60.
So could this be related, your aspirin findings are also very intriguing and really fascinating. And most of the people who get aspirin for prevention of cardiovascular disease now are older. I'm sure you control for age, but so the population who had glioma and aspirin were certainly older people. Is this?
I need to go back and, we probably should look at that, because I'm sure we thought about that because of the age that it's, so we must have controlled for age.
Right.
Yeah.
Yeah, that'd be really interesting.
I know when just a personal, I take it because I also have a family history, but, and my doctor said, well, it's not recommended anymore. And I had to go through a whole spiel about why I still want to take it. And she just said, okay, I don't want to hear about it, or something like that. But, because I feel like our data are strong enough that taking a baby aspirin doesn't cause me to bleed or anything like that. So, I recommend it.
All right. And the interesting also, the difference between male and female, is there also a gene association in this? Or, because the difference is pretty high.
Yeah, it's like 1.4 to 1. There's been quite a few investigations, and Quinn again had done some of this for her dissertation, and we looked mostly at the SNPs, not at others, and there was a little bit of difference between some of these, and there's been some other studies of sex differences. It's really hard to figure it out, in terms of identifying if there are different genes that are associated. But hopefully with the data that we'll have in the next set, we might be able to look at that a little bit closer. Because we didn't do whole genome sequencing before of the tumor, and then we have, other types of sequencing data. So as I said earlier, technology drives the research.
Right. All right.
Thank you so much. If there's any more questions, I'm happy to answer. And my email is mbondy@stanford.edu, so I'm happy to respond to questions later.
Okay, great. Well, thank you so much for your wonderful presentation.